Okay, hello. All right. So I was asked how to make RNA probes, specifically intronic RNA probes. So that's what we're gonna cover today. So here's the quick tutorial. Um, let's just choose a gene that is, yeah. So go to the UCSC browser, go to the fly genome. It really doesn't matter which release you use, although 6 is the most recent one, so it's going to be the most complete, so probably the best one. Go to the gene that you want to make the probe for, and then we're going to do a couple of things. But really quick, I just want to mention that the reason that we want to do an intronic probe is because we want to label the RNA that is very recently transcribed, right? So we don't want to, for example, label a bunch of maternally loaded mRNA that might be, floating around the embryo. We want to actually see what's being transcribed right now or very recently transcribed, pre-splice. So what we want to do is find a region of intron, right? So intron is so the way that these, these RefSeq gene annotations work is introns are the lines with the arrows, and these solid blocks are the exons, right? So, and then these are the 3' and 5' untranslated regions. So, what we want to do is pick a region of an intron that's in all of the known isoforms, right? So that's really easy for this gene because, right, both introns are the same for all three versions, and so we could pick anywhere in here. However, for the best chance of seeing as much signal as possible, right, because presumably the least amount of splicing is going to be while the RNA molecule is still attached to the RNA polymerase on the genome itself, right? So we would want to choose a region close to the 5' end of the gene, right? So it's going to be attached to the RNA polymerase as long as possible. You could also do it on the 3' end, but you're going to get less signal, hypothetically, as a potentiality. So what you're going to want to do is go ahead and click up here above these numbers and hold your mouse clicker down, right? So you can see I'm selecting a region right there. Also, the probe sequence size apparently is optimally 1500 base pairs. However, I've had probes work perfectly fine that were only around 800 base pairs, and I've heard of people using probes as low as 500 base pairs. So if your gene is really small or something, don't worry about it. It's still worth trying. But 1500 base pairs is a lot, so you should get more signal. And yeah, so that's what we're going to do. Now, instead of just choosing exactly 1500 base pairs, we're going to want to be able to play around and find, an optimal region. So let's go ahead and select twice as much, 3 kb. And then click zoom in the UCSC browser and click on view DNA. Okay, actually one thing, let's go back. So we're seeing that this DNA sequence is in the sense direction with the sequencing of this chromosome, right? So this is the forward direction of the gene. The forward direction of the gene and the forward direction of the sequencing of the chromosome, annotation of the chromosome, is the same. So we're not going to reverse complement it. We're just going to click DNA, get DNA. But if it was in the other direction, I like to push get the reverse complement so that whenever I copy this and paste it into a new analysis, or a new gene file, whatever, I know that I always put everything in the direction of the gene. So it's in the genic direction. This is the 5' end of the gene. This is the 3' end. Right? And then label it HMX intron 1 fragment, maybe. Yeah, sure, why not? So now you can see this is what it looks like. We've got the sequence in here, and I've got this set to show me the unique cutters of restriction enzymes, right? And if anybody needs more information on how that sort of thing works, I can do that in another one of these videos if you want. Just let me know. So the plasmid that I'm going to use as an example is the one that I like to use the most in my current lab to make RNA probes, which is just PGEM. This version of PGEM. But you can use whatever you want, PUC, whatever, doesn't matter. As long as you've got your RNA polymerase, as long as you've got your promoters, then you should be fine. And the promoter that we like to use is T7. So it's good to know that that is there. So let's take a look at the multiple cloning site. So we want our probe sequence, our probe's DNA sequence to be between the T7 promoter and the SP6 promoter. Well, okay, mostly we just want it downstream of the promoter that we're choosing, right? And directly downstream. And so we've got the two options here. And again, like I said, I'm going to choose T7, but you could choose SP6 if you have SP6 RNA polymerase in your lab, whatever. But anyway, so what we need to do is select, restriction enzyme sites that will allow us to ligate our probe sequence into this multiple cloning site. So these are the enzymes that we have available. You're going to need to also make sure that you have the enzyme itself in your lab for use when you're actually ready to do the cloning. So keep that in mind. So we're going to go back here, okay, and we're going to look for some of them, right? So for example, oh shoot, sorry, wrong thing. Let's go back here. So for example, EcoR1, KPN1, BAMH1, XBA1, and HIND3 are all very, very common restriction enzymes. So let's look for those. Not here, but actually, if you're using SnapGene, this is very easy. In other applications, you'll have to figure it out. But in SnapGene, you can go to enzymes down on the bottom, click non-cutters, and then just look for those ones. This is the way I find is easiest. So we can see EcoR1, KPN1, XBA1, and HIND3 are all non-cutters for this sequence. So we can choose any part of it and use any pair of those restriction enzymes for our probe cloning. So I remember that I wanted it to be, so now you'll remember that we wanted it to be close to the 5' end of the intron, right? So let's go ahead and just try to make a probe kind of, here-ish, about 1,500 base pairs, right? So something like that. So what we're going to do is take a look at, and again, SnapGene makes this very, very easy. We're going to take a look at predicted melting temperatures of 20 base pair regions here, because we're trying to figure out where to put primers to be able to amplify the sequence for the probe from the genome. And these are very, very low. Like you can see, this is a 40% GC content. That's a very, very low GC content. I want something closer to, I don't know, 50 to 55. I find that higher temperatures when you're annealing in your PCR to the entire genome, a little bit higher temperature tends to give you more specific bands, if that makes any sense. So I generally tend to look for at least a 53. Okay, yeah, you know what? This is actually pretty good. So 50% GC content and 57 to 58 C here. Right, so that'll work. Then you just go up here. Oh no, I cut off the top of this. But anyway, up here there's a toolbar, and the toolbar includes one that says primers. So you click that, you click add primer. Then this will come up. We want this to be the top strand, and then we'll label this HMX Intronic Probe F for forward primer. And we're going to insert that into there. Now we're going to go ahead and grab the DNA and slide down until we're in the neighborhood of 1500 base pairs, right? So that's where 1500 base pairs is exactly. So we want a 20 base pair sequence here that's roughly the same temperature as this. So this was 58. So we're going to look for a 58 here. And, you know, just somewhere in this neighborhood. It doesn't have to be, you know, plus or minus 100 base pairs. And also plus or minus a couple degrees, and you're going to be fine. So I can tell that's a little low, so I'm going to slide over a little bit to some more G's and C's. Okay, that's too much. So let's cut that off a little bit. Go over here. 59, we're on the right track. I think this will be it. I think we can do better. Let's go here. Okay, anyway, I mean, there are also functions that you can use. I think even SnapGene has them built in, where you can ask it to find you primers, but I don't really know how to use them, and this takes a couple seconds normally, so it's not that big a deal. Let's try this. Let's see if I can get the exact time. Okay, I can't. So this will be fine. So let's go ahead and do this one right here. 20 base pairs. And if you have a lot of trouble, like sometimes, especially in introns, you can end up getting sequences that have a whole bunch of, like a big block of G's and C's, or a big block of A's and T's. And so it's really hard to find, you know, a region, you know, an actual primer sequence that works on both ends. So you can always, you know, worst case scenario, you can actually just change the lengths of your primers as well. But I like to try to stay the same length and roughly the same temperature. So I'm going to go with that. Add primer. Bottom strand this time. 56. I actually don't like that. So I don't want to be 2 degrees different. I want to at most be 1 degree different. 2 will probably be fine. But I just want to check if I can do a little bit better than that. Yeah, this will be better. 59. You know, that's better. All right, so this is HMX Intronic R. Now that we have those, and again, the temperatures are pretty high, which is good because if you think of the way that PCR works, right, you should, higher temperature should yield less off-target PCR products. right? Because then mismatches are not going to anneal as easily. So let's go ahead and just go to blast. Blast really quick. Okay. And then we're going to go to primer blast. And we're going to paste our 2 primers in here. It's in Snapchain, it's easiest to do it this way. Okay, drop them in there. Then you're going to want to check here. You're going to want to do representative genomes, okay? Right, because we're labeling introns. So if you choose RevSeq mRNA, it's not going to have the introns. And you're going to want to do Drosophila in this case because that's what we're designing the probes for. And then we're just going to push get primers. You can also change these parameters if you feel like you need to for some reason. But in general, this has always worked for me. So my prediction is that it'll basically say that there are a couple off targets that are very unlikely. And we just have to wait for this government website to load. And yeah, so here's the result. So, oh, look at that. HMX is the target and 1513. So basically what we're expecting, and then some off-target ones. So what we specifically want to do here is look for ones that are relatively close to 1513, because those are going to be hard to, like, if there's, some band that's a perfect match, right? But it's like 4,500 base pairs or something, right? That's obviously one, like the PCR is going to limit that one, but when we run it in the gel, it's going to be very easy to find which band is the right band, right? So like looking through here, I would say the only one that's really concerning is this one right here. And that's because this one's only 200 base pairs difference, which, that could be very difficult to actually detect in our gel when we're running it during the cloning process. However, if you look at it, like, I mean, that's 5 mismatches and 4 mismatches. 4 mismatches. I mean, it's not even the same size. So I'm gonna, if I was making this for my actual project, I would go ahead and make it. I'm in a sequence anyway, but my prediction is that this won't happen. Like, I would have to lower the temperature a lot to get this to happen. So I think these are a good primer pair. So let's go back to here. So remember, we looked at EcoR1, KPN1, XBA1, and Hind3, and found that they're all non-cutters in that larger sequence, which means, of course, that they are non-cutters in this smaller sequence, which is our probe sequence, right? And so now we can put any pair of those here on the ends of our primers in order to be able to do the ligation into this plasmid. And so, for example, let's go ahead and we'll choose EcoR1 and Hind3. And so what we're going to want to do is make our primers basically, let me, wait, where did it go? Here. So our primers, I like to go ahead and put them in here like this. So at the beginning of this, we want to add some garbage, right? And the reason for that is that the ends of DNA are fragile, so you want a couple base pairs that are just sacrificial, so to speak. And then between that sacrificial piece and your primer, you're going to want to put the enzyme sequence that you're going to use to cut the ends of your PCR product and the middle of your destination plasmid with restriction enzymes to do sticky end ligation. Well, in this case, we're doing sticky end ligation. So, but you're gonna, for this purpose, there's no reason not to do sticky end ligation. So, so let's go ahead and choose our enzymes. And for this case, we're just going to use EcoR1 and HIND3. And so now we need to make it correct so that we use our RNA polymerase and get the RNA that we need to label the well, not the mRNA, but the intron in this case, with our probe. And so that means that we want to transcribe the sequence in the opposite direction of the gene, right? So that it's producing complementary RNA to the RNA that's being transcribed in the animal itself, in the embryo, right? So in our forward primer, we're going to want HIND3, and in our reverse primer, we're going to want EcoR1, so that the gene ends up pointing towards T7, so to speak. So the HIND3 sequence is this, and so you'll put that here. And when you order from IDT, this is what you're going to, or wherever you order your primers from, this is the sequence that you're going to want to get. Okay. And then you'll do the same thing for your reverse probe. Or your reverse primer. Put some sacrificial garbage base pairs there. And the EcoR1 sequence between. Okay. And then you can do your PCR with genomic DNA, do a ligation, enzyme digestion, digest your PGM as well, and then do your ligation, and you should get this, right? Your gene, your probe sequence in the opposite direction of the endogenous transcription, so that you can use T7 to transcribe it, right? And don't forget to use HIND3, which is what we chose on the forward primer, right? Don't forget to use HIND3 to cut before you do your in vitro transcription, but that's a topic for another time. Have a nice day.